drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. You want to just start off by telling everyone a little bit about who you are and what you study?
Sure. Yeah. Name's Gary Patti. I'm a professor at Washington University in St. Louis. Actually wear a few different hats. I'm in a couple different departments: chemistry, medicine, and genetics. I run a relatively large lab. We study cancer and metabolism. I also serve as director of some cores at the university, the clinical research core, as well as some metabolomics and proteomics cores. And then lastly, I'll just mention that I'm also a CSO of a company that does contract research services for different omics, multi-omics technologies.
Mm-hmm, mm-hmm. Yeah, so you've got quite an interdisciplinary lab. You do a lot of stuff that's related to metabolism and metabolomics. You do a lot of stuff related to cancer biology, which will probably be our focus here today. We want to get to a paper that I thought was very interesting and very relevant to a lot of people, out in the real world, which has to do with the relationship between sugar metabolism in particular and tumor growth. Before we get there, I want to cover off on some basics on cancer biology and sugar and fructose metabolism. So, I'm somewhat familiar with cancer biology. I'm a biology guy by training, but I was never a cancer biology person. And sort of the cartoon that I've always had in my head, or I had when I was like a student in college and stuff, is that, cancer is a cell, cancer cells are cells that have escaped the normal cell cycle regulation and they're growing uncontrollably. And that is largely often a genetic thing. So, I learned about oncogenes and tumor suppressor genes. And so the idea was cancer is typically a series of mutations in specific genes that allow the cell to break free of the cell cycle. Can you just give us a general sense of, are most cancers, or is the average cancer, whatever that means, is it largely a matter of mutations in genes, or can cancer arise through non-mutation mechanisms?
It's a really great question and a good summary of the historical. There's a little bit of history and how our understanding of cancer has evolved over the last 100 years or so. And I won't impose an age upon you, but I think depending on when you got trained and when you learned about some of this stuff, different items and different aspects of cancer biology were emphasized.
Mm-hmm.
Some of the earliest discoveries associated with cancer were not based on genetics, but actually were metabolism. If you go back about 100 years ago or so, there was a German scientist named Otto Warburg, who was really the first person to think about cancer from a metabolic perspective. And he noticed that cancer, sugar metabolism specifically, which we'll get into more here in the next few minutes, was altered in cancer. And so his definition of cancer, actually, a century ago, was that cancer cells have altered metabolism. And he actually asserted that a certain piece of cancer, an organelle inside cells called mitochondria, that is sometimes known as the powerhouse of cells. It's not the only place where metabolism occurs, but at least we teach the undergraduates that that's where a lot of the important metabolism, certainly where most of the energy in a cell is made. He actually argued that what causes cancer is not oncogenes, as you described, but actually failed mitochondria. And that it was that alteration in metabolism that was the cause of cancer. Now, we know that's usually not the case. Most cancers have completely functional mitochondria, and in fact, they need mitochondria. That's something that our perception has evolved over the years about cancer. But as genomics evolved, there is in many fields, cancer fell victim to sort of getting obsessed with molecular biology and genetics. And
as a consequence of what the tools were that emerged.
Yeah, exactly. Yeah. I mean, I think the Human Genome Project, played a big role in that, and the excitement of being able to do sequencing. I think, and rightly so. I mean, it was an exciting time. In 30 years, we'll look back on today and say, oh God, they were so, they were obsessed with whatever we're doing today. Yeah. So I think that's just how science evolves. But there was we learned that genes, mutations in certain genes were sufficient to cause cells to behave abnormally. And specifically, not just abnormally in some way, but they lose their ability to regulate proliferation. So they proliferate uncontrollably. And that's really one of the main driving characteristics of cancer cells, is that most normal cells know when to stop growing and stop turning into new cells, but cancer cells don't do that. They proliferate uncontrollably. And so genes are at the basis of that, and they cause that. But exactly how they cause that, I think, is the really interesting question. So I think what you say is totally correct, but it's only a piece of the equation. How does an oncogene allow a cell to proliferate uncontrollably, where a non-transformed cell that doesn't have that oncogene doesn't do that? How?
And I guess if you just think about it in sort of common sense terms, it would almost have to be true that in some way, whatever these genes are that might drive certain cancers, they would have to affect metabolism, because almost by definition, cancer is going to have an altered metabolism because it is uncontrolled growth.
Yeah, it's actually amazing how long that I totally agree with you. But there are several decades where metabolism was backburnered as part of cancer. I mean, I think it's fair to say that cancer is a metabolic disease. There are certain hallmarks that classify cancer, and altered deregulated metabolism is one of the hallmarks of cancer. So it's not specific to pancreatic cancer, or lung cancer, or brain cancer. Altered metabolism is something that most cancers across the board share.
Yeah. I want to ask a somewhat vague question, but it's an important question. And anyone who's not a cancer biologist, like I'm not, might wonder this. So obviously there are different forms of cancer, in the sense that the cancer can arise in different tissues: pancreatic cancer, brain cancer, whatever. But how much how much diversity is there in terms of the fundamental type of cancer it is from a cell biological standpoint? Do most cancers, is it sort of the same basic phenomenon, or is there a lot of variation where two different cancers can behave cell biologically very different from one another, apart from the fact that both are unregulated cell growth?
Yeah, amazing question. And one that I think that we're still trying to figure out. Certainly the tissue of origin is what you described. So some cancer cells might be derived from pancreatic cells, or, what hepatocytes, or whatever cell type they originate from, plays a role, because those cells are already programmed to operate in one way. And so certainly you see diversity if you look at tumors in different tissues. But what's really interesting is that there's also quite a bit of diversity in those same tumors. So it's not just diversity between brain cancer and liver cancer. It's actually, there are lots of diversity in brain cancer.
Right.
And so I think certainly the tissue of origin plays a big role in how provides really a starting point for how the cell is going to thrive.
Mm-hmm.
But the other thing that plays a major role, and this is a good segue into some of the other topics we're going to discuss, is the environment.
Mm-hmm.
Because cells to proliferate, so just to really provide context for what we're talking about here, one cell has to turn into two cells.
Yep.
So a cell has to replicate its contents. And to do that, it requires a lot of synthesis. You have to rebuild all of the protein machinery, you have to rebuild all the lipid membranes, you have to rebuild all the genetic material. And that takes a lot of biomass, and you have to build all that stuff from something, and it takes a lot of energy. And so the question is, what nutrients is a cell going to use to support that anabolic process?
Mm-hmm.
And what's remarkable about cancer, I think, that's totally fascinating, is the versatility that they have and the plasticity that they have in the nutrients that they can use. It's rare that they are limited to one nutrient. We're going to talk about sugar today, and they love sugar. Most cancers are addicted to sugar. And you might think, well, if we remove sugar, and we've tried this experiment, starve cancer cells of sugar, then does that prevent them from doing that? In most cases, the answer is no. They just move to a different nutrient. So there is a lot of flexibility. They don't always operate. Yes, they start as the tissue of origin. They seem to start in that way. The other thing that happens is that tumors can metastasize. So you can imagine that a tumor starts in, the lung or the breast, and then it metastasizes to the liver. So now you have cells that originated as breast cells, but now they're in the liver in a totally different environment. And so it gets kind of interesting. Do they start behaving more like primary tumors that recur in the liver, or do they retain their program that originated in the breast, but conduct that in the liver? Mm-hmm. So you get those kind of interesting questions too, because cancer is mobile.
Mm-hmm. So my understanding is we're basically all getting cancer at some baseline rate, but most cancer cells that emerge get taken care of by the immune system. They're detected, the body notices that they're abnormal and we don't want them, and then we get rid of them. But obviously in some percentage of cases, that those checkpoints, those mechanisms can't get rid of the cancer, and then we get full-blown cancer. When we think about cancer generally, are there sort of certain phases or parts of the life cycle that we can talk about where so obviously cancer can be initiated, but if a cell becomes cancerous, that doesn't mean we're going to get cancer, because the immune system might get rid of it, and it often does. What is that transition like, where you go from a cancer cell you get that first cancer cell and it gets past the normal mechanisms the body has to detect it and becomes, say, a full-blown tumor?
Yeah. So from a metabolic perspective, I think it's a really fascinating question. And it's proven to be somewhat difficult to study, because usually what we're talking about there are small numbers of cells. And if you think about the ways that we generally study cancer, most of those things are subclinical. Like you can't see
you don't see that first one. There's no symptoms.
Exactly. There's no symptoms, and you can't detect it on we often use something like a PET scan. I think most people are probably familiar with PET scan. So essentially what you do is you give patients a radioactive chemical, and then you put them in a scanner, and you can detect the uptake of that chemical. And unfortunately, if you have a small number of cells, one cell or a handful of cells, they don't show up on those kinds of tests. So it's proven to be very difficult to study these processes, and the way in which we typically do it is fairly artificial. So we can study them in things like culture, in cell culture.
Yeah.
We put them in a dish, and you could do things like
Hmm.
irradiate them or cause
Yeah. Yeah.
initiation by giving them chemicals and see. But how it translates to the body, I think that's an exciting area
Yeah.
where we keep pushing. Now, the other thing you bring up, which is incredibly important, is the immune system. And that's an area where I think we've seen enormous development and progress over the last 10 years, where it's not just because you're right, the immune system plays a major role in trying to keep things contained and controlled. But it doesn't always work. I mean, a lot of people get cancer. And so, I think one of the really exciting things that's been happening is can you tune the immune system in such a way to make it more aggressive, more active, to go after some of these tumors. And that's when you start getting into the immunotherapies, using the immune system to try to sort of put the immune system on steroids
Hmm.
to go after this
Hmm.
stuff. And it's been pretty effective in a lot of different. It's not obviously not perfect, but a lot of progress in that
Yeah.
space.
So it sounds like the sort of basic problem here is you can study the origins of cancer. You can look at the birth of that first cancer cell, the early phases of, going from one cell to two cells to a small tumor. You can study that in a dish in a artificial setting, which is amenable to experimental interrogation. But obviously, that's removed from the natural context of the whole animal that we ultimately want to understand it in. Actually, understanding those early phases of cancer in a whole animal in vivo situation is tough, just for the very reason that it's hard to spot it when it happens. It's
Yeah.
small and difficult to detect, and we don't really see these things in animals or humans until they're in a much more mature phase.
Yeah, yeah. I could give you one sort of. I think what you said is generally true. There are, ways, innovative ways that people are coming up with to try to study this, and I can just give you a little bit of a technical example if it'd be
Mm-hmm.
of interest to you. And that is a way that we've been trying to tackle this question using a model organism called zebrafish. And zebrafish are interesting model systems for people that aren't familiar with them. They're about the size of goldfish, and they are aquatic, and they present some advantages. They, they're they're translucent, so they're easy to study. And what we do, if you put a oncogene in fish, you can put a certain genes like we use BRAF V600E, which is a mutation that commonly occurs in various forms of skin cancer. And if the fish have that, just like some humans that have genes, like you might be familiar with, say, the BRCA genes, you have a higher incidence of developing cancer. The fish, too, because they have this gene, will have a higher incidence of developing cancer. And so what we do is we genetically engineer the animals to have this gene, BRAF V600E. And then when the gene gets expressed, we use that expression to drive the expression of GFP, green fluorescent protein. And what that does, particularly in this unique system of a zebrafish, which, like I said, is translucent, we can
Mm-hmm.
put them under a microscope. And when the cell turns cancerous,
You
at the
see it.
single cell level, it turns
Yeah.
green.
Yeah.
So we can do things like start asking questions, that we've figured out a way to make it more visual, so that we can study it more quantitatively at higher detection limits.
Yeah.
And we can do things like ask, how does sugar influence initiation? Or how does sugar influence the development, those early stages of development, before you get a clinical sized tumor? Does diet play a role there? I think
Mm-hmm.
those are really interesting questions that, like I said, we're just starting to really develop some tools that are allow us to dig into some of that stuff.
Mm-hmm. So again, high level, I would imagine that when a cancer, so there's the birth of a cancer cell, most of them aren't going to live long past their birth because the immune system will take care of them. But when they do get past that, does that involve there must be some kind of evasion technique that the cancer cells use? Are they able to camouflage themselves or just avoid, how do they avoid detection by the immune system to really get going as tumors?
Yeah. Well, there's a couple of things that are involved. There's also intracellular repair mechanisms, that can, the immune system isn't the only ways and the things that need to be evaded.
Mm-hmm.
There's other DNA repair type systems that can come into play as well. So fortunately, we have a couple of mechanisms to try to catch these things. But the other thing that starts to be really interesting is signaling. So cancer cells at a certain level start to really when you get enough cancer cells, you start to develop a tumor. And when you have a tumor, at some point, it starts to integrate into the physiological system of the host. So if you think about your body, you have a bunch of different tissues, a bunch of different organs. You have liver, kidney, lungs. And when you have a small number of cancer cells, they just operate kind of independently in their own world.
Yeah.
But when they get to be a certain size, what starts happening is they start integrating into the physiological system of the host. So they start communicating and signaling with other
Mhm.
tissues.
So they start releasing, detecting molecules coming from healthy tissues to literally sort of integrate physically with the healthy
Exactly.
tissue.
Yeah, they start engaging in basically biochemical exchange. So they
Mhm.
release molecules, they go to other tissues, and that changes, reprograms the tissues. That other tissue might send molecules back. So they have this intercellular communications and exchanges, and that gives them a certain robustness that makes them much more difficult to try to combat. The other thing that tends to happen at certain limits is like we think of vascularization.
Mhm.
So you get sprouting where you actually get more blood vessels that come in and start to feed because as you get more and more cells, you need more and more nutrients, and the blood supply, the nutrients are primarily derived from the blood, and you need blood to do that. So you'll start to actually vascularize the tumor. So they start they start basically manipulating our native responses and using those in pathological ways. And I think, in my opinion, that's one of the most interesting aspects of cancer right now is that we're starting to appreciate that. And some of these things we've known for a while, but it's becoming, I think, even surprising on the context of some of the more historical stuff. And that is that they're really able to hijack a lot of the systems that are already in place and use them to their advantage.
Yeah. So I mean, naturally, you would think so. If I mean, by definition, these cells are growing too fast, so they need to fuel that growth somehow. They're going to need to instigate a remodeling of, say, the vasculature, so they can get that extra blood flow that they need to support that. They're going to need to sort of suck up extra nutrients compared to what a normal cell would, and that would somehow have to involve, I mean, not only sort of building just the physical capacity to do that, like the blood vessels that will inject all those nutrients, but they probably have to signal to other cells to sort of, I would imagine that like our cells are normally set up with mechanisms to say like, the nutrients have to be apportioned according to the needs of all the cells, and I would
Yeah.
imagine there's natural mechanisms that prevent one cell from getting out of control, and somehow the cancer cells have to circumvent those things so they can take more than their fair share, basically.
Yeah, exactly. I mean, I think that's exactly right. I mean, if you think about just nutrition as a whole, you put food in your body,
Mm-hmm.
and that food has to be allocated
Yes.
among all of the trillions of cells in your body. And how does that actually happen? Like who decides who gets to eat what?
Mm-hmm.
And it's actually remarkably complex and symbiotic. That one cell, the way it's just totally amazing. I mean, metabolism is just so freaking cool because what happens is that some cells say, okay, well, I'm not going to use these nutrients because we need that nutrient for, say, the brain. The brain is highly specialized and it needs nutrient X, so we're not going to touch that. And then these other tissues over here, they're not going to use this nutrient because this. So everybody gets sort of assigned certain ways in which they should biochemically behave so that everything operates correctly in a holistic fashion.
Yeah.
And what happens with cancer is that they just sort of disobey all those rules. They come in and shake everything up. And not only do they themselves use nutrients in a way that is flies in the face of the whole physiology, but they reprogram other tissues to use nutrients inappropriately. So it just really throws the whole thing out of whack.
Yeah. Yeah. And okay, so you know where we're going to go with this particular fructose paper we're going to end up talking about. But
Yeah.
before we get there, but with that in mind, when we think about certain types of cancer, tumors, I'll let you decide which ones are the best to talk about. But the tumor microenvironment that gets created to support this unchecked growth that is cancer, what are some of the hallmark metabolic changes, or is that even a good question, that support that growth? And I guess what I'm really asking there is, Is this typically a process that's going to rely on heavily on glucose metabolism? Is it going to be a mitochondria thing? Is it going to involve, using glycolysis more or less? How does a cancer cell oftentimes switch its metabolism relative to a healthy cell in terms of the bioenergetics?
Yeah. Yeah, there's a ton there. I'll start to tackle some of that, and you can steer me along
Mhm.
as I probably spend the next hour trying to answer that one question. So how does a cancer cell, so one of the things you said is how does a cancer cell itself reprogram its metabolism
Yeah.
in the face of this anabolic burden that has to replicate? And that alone is an incredibly interesting question, and one that's actually puzzled cancer biologists for over a century. It seems like it should be pretty easy. You'd say, okay, well, the cell needs to replicate, so it should just dial up all the pathways. Everything should be up. It should take up more sugar. It should take up more of every nutrient and just
Yeah.
ramp up every metabolic pathway. And that's actually superficially not what seems to happen. So if you think about, I'm going to try to break metabolism down extremely simply here because
Yeah.
I know a lot of people probably aren't into the weeds, aren't as excited about this topic as I am. But when glucose comes into a cell, it can be really metabolized in two ways.
Yep.
It can be metabolized without oxygen, which converts it into a molecule called lactate. And when you do that, you only get 2 ATP, which is the currency of energy in a cell. But alternatively, the glucose can be metabolized oxidatively in mitochondria, totally break it down into CO2. And when you do that, you get something like 30 to 38 ATP. So it would seem that the cancer cell needing a lot of energy
the second way.
The second way. But that's actually not what happens. Most of the glucose that enters a cancer cell gets excreted as lactate. And so you don't get a lot of energy, and most of the carbon is wasted because it's lactate is a byproduct.
Okay, so a glucose molecule can come into a cell. It can go down. You can metabolize it in the cytoplasm without oxygen and get a couple of ATP molecules, or you can funnel it through a mitochondria, and it's going to do the oxidative phosphorylation thing. You're going to use oxygen, and that you can get a lot more ATP, a lot more bang for your buck that way. You would think naturally, cancer cells got to grow a lot. It's going to probably crank up and use the more efficient way to make ATP, but you're saying that often does not happen.
It does not. Yeah, that's right. So it's like I said, this has been the paradox. This is something that's almost synonymous with cancer metabolism called the Warburg effect. It was observed by this German scientist, Otto Warburg, 100 years ago. And we've been trying to figure out why that doesn't happen. If we asked 100 people would probably say that's what should happen. Why that doesn't happen. And, I think we finally start to understand why, I think we now have a good explanation for that. But the key thing to emphasize here for the listeners that might be saying, well, you just said that tumors outgrow their blood supply. So the reason they metabolize glucose in this way without oxygen is because there's not enough oxygen. So they're doing this because they're hypoxic is the word. And that's a great point. But cancer cells do this in the presence of oxygen. That's really the key thing.
Yeah.
Even though they have oxygen, they still do this.
So in a sense, they don't need to do it this way, but they choose to.
They choose to. And it does seem, and so I think we've made a lot of progress on trying to answer this in the last 5 years or so. And actually what seems to happen is, if I could kind of jump to the punchline, what seems to happen is that the cancer cells take up too much glucose. So the reason that most of the glucose gets excreted as lactate is not, as was once posited, that it's because the cancer cells don't want to metabolize it oxidatively. But as it turns out, the amount of glucose that they take up is far greater than the amount of maximum glucose they can oxidize. So they do oxidize a lot of glucose in mitochondria. It just turns out that they take up so much more glucose than the mitochondria can support that it gets excreted.
So the mitochondria, they're using the mitochondria. It's just that the mitochondria have there's a rate limiting thing here. They can only process it so quickly, and all the excess stuff will naturally just go to glycolysis. And because they have so much glucose, that's going to be a natural side effect. That seems to bring us to the point of, okay, so they are somehow sucking up a hell of a lot of glucose.
Exactly. And that's really since the 90s. That's something that we've exploited clinically. So if anybody's wondering, what is metabolism done? Who cares about metabolism? Why does it matter? The best testament as to why metabolism is important is because this is the main way in which we diagnose, stage, and test tumors, how they're responding to therapy. So I alluded to this earlier, but now that we've provided a little more context, I can give a little more detail. So what you'll do is you give patients a radioactive form of glucose.
Hmm.
And because tumors take up glucose faster than any other cells, you've probably seen these images. They're
Yeah.
called PET scans.
Yeah.
And the tumors are really bright because they take up all of the radioactive
Mm.
glucose, and most of the other healthy tissues don't. And that's a main way in which how we diagnose tumors, that they're there, or try to stage how big they are or how well a person's responding to therapy, as we do this test. Now, the other point, if I could go back to your original question, which was really contained a lot of information, is that when we think about tumors, an important point is that tumors are not just cancer cells. So cancer cells, I would say, are these cells that are, like you said, have oncogenes. They're malignant. We say they're transformed. But if you look at a tumor, yes, it has these malignant cells, but a large portion of the cells are not malignant. Tumors actually recruit healthy cells to their microenvironments, and they abuse them. They're ostensibly healthy, but they're reprogrammed in a way to support the tumor.
Wow. It's
And
like someone who's unwittingly recruited into like an organized crime scheme, and they're just they're just the shop operator, but they need them to run the operation.
Yeah, no, I like that analogy.
Wow.
I mean, the analogy I often use is it's kind of like outsourcing. We said that cancer cells need a lot of materials to build, to build stuff. And just like if you were building a house, you may not want to do everything from scratch. You can go to Home Depot and buy pre-made toilets or pre-made whatever. And that's sort of what cancer cells do. They can rely on some of these other cells to outsource some of the production. And so these other cells then can produce things and ship them off to the cancer cells. And then the cancer cells don't have to start from scratch. They can build from these. It puts a lot of that energy demand and that anabolic burden on other cells.
Yeah.
So you had asked maybe for an example. And I could just give you one example that I think is a really easy among those that are known, maybe one of the easier ones to conceive. And that is breast cancer. So in breast cancer, you have cancer malignant cells that are juxtaposed next to fat cells, adipocytes. And those adipocytes, an adipocyte is a cell that stores a lot of lipid. And lipid is people probably can appreciate, is a really rich source of energy that you can burn for fuel. And what we know happens, actually, not just maybe we can get into this later, not just breast cancer, but tumors. A lot of tumors have a disposition to want to make themselves adjacent to fat. And what we know happens is that these fat cells funnel the lipids that they have inside to the cancer cells, to the malignant cells, so that the malignant cells can use it as a type of fuel or to build things, to build membranes and so forth. So it is literally using the lipid component of these adipocytes to feed the malignant cells.
Yeah. And there's a whole world of stuff here. I've had people on recently, we were talking about things like polyunsaturated fatty acids and their potential role in supporting tumors that originate in colon cancer. And maybe if we have enough time at the end, we'll loop back to metabolic flexibility and lipid stuff. But keeping on the subject of sugar. So, I just want to repeat a couple things for people. So I don't even know if I fully appreciated this. One of the simple, basic things that you said was tumors are not just clumps of cancer cells. It's a whole environment that includes cancer cells and healthy cells that have been sort of recruited into this mass. And so there's normal tissue and cancer tissue in a tumor. The cancer cells themselves, the cells in the tumor, are really good at sucking up a lot of energy. And it sort of alluded, at least, that they can suck up energy. They'll get energy some way, somehow, no matter what. But they often are using glucose because that's what's around, and that's what's going to be easiest in some sense for them to support this unchecked growth. We've mentioned glucose already. Before we get into the cancer stuff more, I just want to give people a bit of a picture on glucose versus fructose metabolism under normal conditions. So glucose and fructose, two forms of simple sugar. What are the key differences there in terms of how cells use these forms of sugar for energy?
Yeah, great question. So fructose and glucose are structural isomers. So what that means is that if you looked at their components, they're exactly the same. They have the exact same collection of atoms. The only difference is that the atoms are positioned in slightly different ways. which is why fructose is different than glucose. As you point out, glucose is we know is key to our physiology. If you think about something like diabetes, hyperglycemia, hypoglycemia, when you go to the doctor, they don't measure fructose, measure glucose levels. We regulate glucose levels with extreme amount of, it's exquisitely controlled. We keep glucose levels in certain ranges. And if you don't do that, your body it's bad. You end up with all kinds of different diseases. It's damaging. Glucose comes from the food. Fructose can also come from the food. In fact, increasingly so. And that whole history there is interesting, because one of the questions that I think is really important, and maybe I'll let you decide if you want to go down that path, but why this trend is happening.
Yeah.
But we've started to consume more and more fructose really over the last 40 years. It's gone up quite a bit. And it's been used in many ways as a replacement of glucose. One of the reasons for that is because fructose, it's actually cheaper. It's cheaper to make, and per molecule of fructose, you get more sweetness out of it.
Wow.
So if you had the exact same amount of fructose and glucose, and I gave both to you, you would perceive fructose to be sweeter.
Oh, wow. So I've known that the fructose glucose ratio has a big effect on palatability and how sweet we perceive things. So I always assumed that the reason that all of these big companies have converged on these very particular fructose glucose ratios of like 55-45 is that they're optimizing for the sweetness that comes from having a little bit higher fructose than you would say get with sucrose. And that's driving increased palatability and therefore increased consumption, which is going to support their business models. What you're saying is actually there's actually like a dual there's a dual thing here, which is not only is it modulating palatability by boosting fructose, it actually is more economic for the companies because the fructose is cheaper.
Yeah, that's right. And actually, you can track some of this stuff back to the embargo that with Cuba back in the Cuban Missile Crisis, when there was a demand to generate. The United States needed to be more self-sufficient, and that's when a lot of the production of fructose started occurring. And if you look back historically, that's kind of when there was a flat line, and then all of a sudden things started to go up. And around that era. Now, there have been some flattening of that curve as of recent, because I think people are becoming a little more, thankfully, a little more conscientious about what it is they put in their bodies. I think that's a message for everyone. We should think about what it is we're eating. So I think, but it's still used massively. I mean, if you walk around and look at the things you're eating, unless you're actively trying to avoid fructose, it's likely that you're consuming quite a bit of it. So back to your question though, about how is it metabolized differently? So when you look at how glucose gets metabolized in the cell, it actually gets metabolized to fructose. You go through, if you look at glycolysis, fructose is an intermediate of glycolysis. Not directly, but its structure.
Okay, so it's an intermediate of glycolysis. So the thing that gets us 2 ATPs per glucose molecule in the cytoplasm, that's necessarily going to create a fructose molecule in the process of generating that energy.
Exactly. Yeah. And so, and when glucose comes in, it's highly regulated. Utilization of glucose is highly regulated. And the kind of off on the control switch there, the control valve, is really high up. When glucose enters the cell, there's a couple enzymes that controls whether or not how much you want to metabolize it. Now, when fructose enters, it's actually possible that it can enter directly, it can bypass that essentially control valve and enter directly downstream, directly into the fructose intermediate. I see. And so essentially what you're doing is fructose has some capacity to bypass some of the regulatory steps that historically that our cells have evolved to basically utilize.
So I see. So on one hand, so two things are coming to mind here for me. It makes sense intuitively that would happen. I mean, if it's an intermediate, you're just skipping the first step because you already have the second step there. But what you're saying is the first part that would normally take you from glucose to fructose is very highly regulated, which would imply that the cell can control and turn things up and down in a very precise manner. if you have excess fructose and you're bypassing that step, the cell's tunability, its ability to regulate how much of what is happening is going to be less.
Exactly. Yeah, that's exactly right. So there's a correlation with the consumption of fructose and the obesity epidemic. We've in around the world, but in the United States in particular, there's an increasing amount of obesity, and some have argued that fructose, the increasing usage of fructose, is associated with that. And it kind of makes sense just from a very high level perspective that if you're putting a lot of nutrient in your body and you can't regulate its utilization, that's going to drive. And we know that when you consume more fructose, that you make more fat. That's definitely happening.
It promotes de novo lipogenesis.
Exactly. It promotes de novo lipogenesis. So we know that's happening. So it all kind of makes sense. So but you're back to your question about fructose. So we've known for 100 years that cancer cells take up glucose. And the question that's emerged in the last couple decades is, well, what happens with fructose? The discovery about glucose was made 100 years ago, but fructose wasn't in high utilization back then. So what happens now that fructose is being utilized? Do cancer cells, should we be using fructose as an imaging agent? And you can imagine that if fructose could be utilized with greater efficiency and without regulation, these are all the things that seem like they would benefit a cancer cell. We talked about cancer cell wants to proliferate without regulation. That's the definition. So it seems like fructose might be a really great nutrient to support that cause. And there has been some evidence, we're going to get to the paper that you're referring to, where we show that, at least in the cancer cells that we studied, that's not the case. But there have been a couple prior studies that had indicated that in some other cancers that is the case, that cancer cells can utilize fructose, and that putting fructose into the body gives the cancer cells access to the fructose, and that escalates, amplifies growth.
So fructose is an intermediate in glycolysis. Glucose can be turned into fructose on the way to creating ATP in the cytoplasm. You can also bypass that by just having the fructose directly sort of plug into that second part there. What about the mitochondria, and what about so when we think about dietary fructose, so exogenous fructose that we are consuming, is there, I guess the basic question I have is can fructose be used to create ATP to the same extent as glucose? Are they as efficient at creating energy as one another?
Yeah, they are. They can both be oxidized in mitochondria. They're both 6-carbon substrates. They both produce 2 pyruvates, which go into the mitochondria and get oxidized. So for all practical purposes, now there is one caveat. And this could come into play. I think it's potentially interesting. You do make a phosphorylated fructose intermediate, and some have argued, so what that means is you're adding phosphate to fructose. And some have argued that because you take fructose up with such high avidity and that you can't metabolize it fast enough, that you end up putting a lot of phosphate on fructose, and that actually sequesters your phosphate. Now, we've been talking a lot about ATP, which is the currency of a cell.
Right.
ATP is got 3 phosphates on it.
Right. So if you sequester all your phosphates, now you're going to be energy limited.
Yeah. So there is some question as to whether or not fructose might be playing a role in some kind of ATP limitation in that sense. And there's this example that people often talk about where they tried to treat diabetics with fructose because they thought, well, if they can't use glucose, let's try fructose. And that ended badly because of at least supposedly because of the sequestration of the phosphate and some limitations that had on the regulation of ATP.
Okay, so now, so glucose, fructose, we've got, cancer cells form. They are good at sucking up energy. Is there anything notable or weird about cancer cells, generally speaking, in terms of glucose, fructose? Are they deviating from normal cells' use of those things in any notable way?
Yeah, that's a really important question, and one that seems like it should have an easy, obvious answer. And that was really the origin of our study. Is that we wanted to take cancer cells in a dish and culture and give them fructose, and we we expected that, and there was literature out there to support it. We expected that the cancer cells would take up fructose and use it like, like crazy,
Yeah.
much like glucose. And so we started this whole paper that you're referring to started with us taking cancer cells and giving them fructose and making the observation that they actually don't utilize it. And we looked at a pretty large number of cancer cells. Obviously, nobody can look at every cancer cell, but we looked at, I don't know, maybe 30, 40 cancer cell lines. And in no cases did we find, now some were a little better at it than others, but it was not able, fructose alone was not able to support the growth of the proliferation of cancer cells. So in other words, if you took out all the nutrients and you only gave them fructose, they really could not proliferate effectively.
But they could with glucose, presumably.
But they could all, yes.
I see.
And not presumably, we actually tested it.
Yeah.
In every case, they could proliferate with glucose, but they could not proliferate with fructose.
Yep. Which is a little weird. As you said, you would have expected them to be good at using either source. You tested a bunch of different cell types in a petri dish, not in a live animal, but you looked at a bunch of different cancers, and none of them were particularly good at using fructose, even though they could all get by with glucose.
Exactly. That's exactly right. And so we started to try to figure out why that was. And the answer proved to be actually quite simple. When you feed fructose into glycolysis, it doesn't just go in directly. You have to modify it a little. You have to add phosphates. And as it turns out, the cancer cells don't have that ability to add the phosphate. They lack that. They don't have the right enzymatic machinery to do it.
So part of the changes that make them a cancer cell have prevented them from having that ability.
So that was kind of where we went next, because like I said, when you think about it, generally speaking, cells have this capacity, but then we started asking, well, is it true that cells in general should have that enzyme? We certainly know that some cells have that enzyme, and in this context, I think it's important to consider how fructose is actually metabolized when you eat it. Because what generally happens, as it turns out, even though we talk about fructose and its potential as a nutrient, most of the fructose, when you consume, even if you're eating, the richest chocolate cake that you can find, stuffed with or whatever cereal or whatever that has enormous amounts of fructose, most of that fructose, or even a Coke, that actually it's got high levels of fructose as well. Most of that fructose is drained from the nutrient components by the time that it reaches the blood. It doesn't make it to general circulation. It gets eliminated by the liver and the kidneys and the small intestine. So most of the fructose does not survive into the circulation.
Why would that be?
Yeah, it's an interesting question. So it seems like what happens is that you get a very small, it's about maybe, I don't know, 1/20th of the amount of glucose right after you eat a high fructose rich meal in the blood. But most of the other stuff gets metabolized by the liver and the small intestine. Yeah. And it turns it into other nutrients that like fat, we're talking about fat. The liver has a special capacity. We know that the metabolic capacity of the liver tends to be much higher than most other tissues. The cool thing about physiology, I think, is that tissues, we talked about that earlier, all tissues specialize. They all have unique metabolism. We could talk about glycolysis. Most of glycolysis happens the same across all cells, but as it turns out, if you really dig into it, there's little nuances in every metabolic pathway between cells. And what the liver specializes in is it has enormous metabolic capacity to break down all sorts of stuff, drugs, nutrients, all the kind of extravagant stuff that we put in our body, the liver gets the blunt end of.
Yeah. So is the reason the liver can metabolize fructose more, is this, are the differences between cells here to do with their ability to import the fructose inside of themselves?
No, as it turns out, we did look at that. The cancer cells have the capacity, and other healthy tissues have the capacity to import. It's actually the hexokinase, the ketohexokinase enzyme that is deficient in other cells and tissues. And also there's a specific enzyme called aldolase B
Mhm.
that's involved.
So-
So those are the
okay.
two enzymes that are required.
Okay, so if I'm hearing you correctly here, most cells can import fructose, but only some cells have a kinase, a particular enzyme that can, let's say, activate the fructose so that it can be metabolized, which would
Exactly.
mean, if I'm hearing you correctly, most cells don't have the enzymatic capacity to metabolize fructose. And so this, I don't want to get too off track here, but I think some people have argued that fructose can be thought of as a dose dependent toxin.
Mhm.
What do you think about that?
I don't think that necessarily changes those concepts. It's just uh, and I do want to clarify. Uh, this is to metabolize fructose sufficiently. As it turns
Mm,
out that even the normal if you pour enough fructose in, even normal enzymes, they're uh, promiscuous, and they can
yeah.
utilize fructose. This is at a very low efficiency. So this is if you want to use it to support and sustain some biochemical function as
Mhm.
for proliferation. It's just not,
I see.
most tissues don't have it.
So is it fair to say that if you consume excess fructose, whatever exactly that means, we'll just leave that kind of open. But if you consume too much fructose, whatever that means, it will be metabolized inefficiently.
Uh, you will get a little bit of inefficiency in the systemic circulation, but most of it will be, no matter how much you put in, the vast majority of it is eliminated
Hmm.
before it gets to circulation. So
Wow.
most tissues just don't have access to
Yeah.
it. The stuff that gets in your circulation, you say, well, if nobody can metabolize it, where does that stuff go?
Yeah.
So eventually it can be broken down, like I said. And there are other tissues in the periphery, like there's a lot of interesting stuff that happens for, say, in like sperm that use it as nutrients. So there are other, it's not that no other tissue, but they're
Yeah.
pretty
Yeah.
limited. And you could just look at the expression of the fructose, the fructolytic enzymes
Mhm.
across tissues, which we did.
Yeah. Yeah.
And you can see in humans that there just aren't a lot of tissues that have the capacity to express those enzymes.
Yeah. So the body is simply not set up to deal with metabolized fructose to the extent that it is glucose.
That's exactly right. It's not set up as the main nutrient like
Yeah,
glucose.
yeah. I wonder if this, I've read too, because I used to be an evolution guy, so I think in these terms. When you look at the genome of primates, the lineage of primates that includes us, um, there's an interesting connection here with uric acid. And I think some people have hypothesized that due to some of the quirks of our evolutionary lineage, we're sort of not meant to have too much fructose, whatever that means, because
Yeah.
because of the uric acid connection here. I don't know if
Yeah.
that's relevant.
No, it's definitely very relevant. And I think the other thing that you said there that we should probably point out, because this is a question that people often ask me, is what is too much fructose? It
Yeah.
certainly doesn't mean no fructose. I mean, there's
Right.
fructose in fruit.
Right.
So we're talking about here the excessive levels of fructose that are in, if you're having a big gulp with lunch and a big gulp for dinner, and you're also eating chocolate cake and cereal.
Mhm.
We're not talking about eating a pear
Right.
or something like that. I mean, there is fructose because the sort of extreme version of this is somebody says, okay, I'm never, especially as we get into the cancer stuff, people say we're never going to eat any fructose,
Yeah.
and that's not. I just want to make sure that that's not misconstrued.
Right. Right.
It's the excessive amount of fructose that causes the issue, and we can talk about that in a bit. Why it has to be large amounts. Exactly what is it? It's a dose dependent thing. It's not like
Right.
this level is safe, and then if you
Yeah.
7.2 is safe and 7.3 is
Yeah.
dangerous.
And maybe to anticipate some of where we go with this, a lot of people get confused on questions like this. Because I've heard this an argument put forth many times. It's like, well, sugar's natural, sugar's in everything. Our body's supposed to have glucose and fructose to some extent. And when you look at high fructose corn syrup, the most common type, I think, is the 55-45. People will say things
Yeah.
like, well, it's only 55-45. Sucrose is 50-50. That's a trivial difference. But when you think about the big gulp and the Dr. Pepper on top of the like these
Yeah.
things accumulating meal by meal over decades, those seemingly small differences can really add up over time.
Absolutely. And I imagine that if you ate enough in whatever form, it would be the same result.
Yeah.
So changing that, the idea of just going to cane sugar and all this goes away, I think that doesn't make sense to me. Maybe some can make arguments for it, but I don't think that's very logical.
Okay. So, anyways, back to this paper. You tested a bunch of different cell lines, cancer cell lines, and they were not using the fructose for energy.
Yeah. And
What?
and I want to stage that and contrast that to another finding that we had that was really puzzling. So the before we even did that experiment, what we did is we took mice that had tumors,
Mhm.
and we put them on high fructose diets. And this would be the extreme version that we're again not eating a pear a day. We're
Mhm.
talking about eating, the big gulp and the doctor, just like you
What,
said.
what did you literally give them?
Literally, so there's a couple things we did. We either gave them fructose by itself, or we gave them high fructose corn syrup. We either mixed it with their chow, or we put high fructose corn syrup in the water, which essentially is sort of like turning their water into soda.
Yeah, yeah.
And so then they consume it avidly.
Mhm.
And so we gave them these diets and quantified how much they took, and we put what you can do is you can transplant a tumor into an animal.
Yeah,
Maybe you
healthy
put
animal.
a healthy animal, so it's totally healthy, no cancer at all, no oncogenes, nothing like that. And you take cells that you grow in a dish, cancer cells that you grow in a dish, that are transformed, and you take a few 100,000 of them, and you use a very carefully transplant them in an animal. And then over time, that those cells will start to proliferate and grow, and the tumor will get bigger and bigger and bigger and turn into a big mass. And what you can do is you can measure how quickly that happens. And so what we did is we transplanted the same number of cells into healthy animals, and then we put them on various different diets and asked whether or not if you feed these mice diets that are high in fructose, if that leads to faster tumor growth.
Mhm. So if the metabolic context is different, that's going to affect the rate of tumor growth, and you guys can measure that.
Exactly. Yeah. And so what we found was is that when the animals consume uh, excessive amounts of fructose, the tumors grow faster in all cases. We looked at all sorts of
Mhm.
tumors, breast tumors, melanoma tumors. One, you go down
Yeah.
the line,
And
and in all cases,
is this like a dose dependent thing? 10% more is 10% faster growth or something like that? Or is this like you have to have an incredible amount before you see an effect?
yeah, we didn't really do the dose dependent. I mean, those are good experiments, and we probably should go and do those. We were just really interested initially if you were kind of at the extreme end of
Mhm.
fructose consumption,
Mhm.
would it have a difference? And we saw in some cases, I mean, the tumors grew 4 times as fast.
Wow.
So just to give you a sense, we're not
Yeah.
talking about a 10%,
Yeah.
5% difference. I mean,
Wow.
these are really shockingly large differences.
Yeah.
So we did that first, and it said it all kind of made sense, in the backdrop of what we said. Fructose looks like glucose, bypasses regularly, kind of made sense. But then when we took the exact same cells that we transplanted in the animal, and instead of putting them in the animal, we put them in a culture dish and we gave them fructose. They didn't use any of the fructose.
Huh. So the cells can't use the fructose themselves, but
Yes,
in the context of a whole animal, the fructose is somehow making a difference for those cells.
exactly. That's exactly right. And we were stumped on that for a few years, trying to sort out exactly what was happening, and exactly why, because that seems like a paradox, right? Those two findings don't seem consistent. And so that was really the backdrop, I
Mhm.
think, and that's why the
Yeah.
paper, I think, what we tried to explain.
Yeah. And before we get to that explanation, I just want to ask one more about the last experiment you described. So you're giving those mice a high fructose diet that was very high in fructose. I know this is mice, humans. It's not a one-to-one comparison, apples and oranges. But caveats aside, is this within the realm of what some humans might be consuming, or is this
Oh yeah.
totally extreme?
No, no, no. It's in the, I mean, this is, and we, in some sense, it was, the animals could consume what they wanted because
Oh, okay.
and you've had some really interesting podcasts, looking at addiction and how food influences our brain and how we want more of things. So, that all comes into play here too.
Right.
But the animals could consume the diets as they wished. So we put the high fructose in the water. We didn't infuse them with the fructose.
Right.
We
You're
did
not
actually
force
do
feeding them.
we did do some experiments where we infused them, but for the vast majority of what we did, yeah, we just gave them access to it. And just like, if you gave me access to cake and didn't tell me it was bad, I'd eat a
You'd
lot of it.
naturally, yeah, yeah.
Yeah, because it is yummy. And so I think that's basically what happened. So it's not physiological, it's super physiological in the sense that we didn't force feed them, amounts more than they wanted to consume.
Yeah, yeah. So the animal's choosing to eat as much as it wants, which is, you know, at a coarse grain level, this is not unlike the natural situation for a lot of humans. We're in an environment where we can eat as much fructose as we want, and a lot of people just do.
Yeah. And I want to make another point here because this is actually really important. We did our study in female mice.
Mm.
Everything we did was female. And one of the reasons why that proved to be important is because you might say, well, if you give the animals a lot of fructose and they're consuming all this fructose and they get obese, we talked earlier about how fructose can lead to obesity, or they develop diabetes, then maybe some of what you're observing with cancer is related secondary to some of these other effects. But what we did verify in these experiments is that the animals don't gain weight relative to the control group, and they don't develop insulin resistance. And for whatever reasons that we probably shouldn't get into here, but very interesting, that the female mice is recalcitrant to that compared to the male mice.
Really?
So they're a little more. It turned out initially we started these experiments looking at cervical cancer,
I see.
which is a female cancer. So we got a little lucky,
Yeah.
fortuitous in that sense. But it turned out, yeah, we tested all this in the female mice, and so it is not because they get fat
I see.
or obese.
Yeah. So to clarify, are you saying that you guys chose to use female mice in this study because you wanted to focus on cancer, not have the confounds of insulin resistance and obesity, and you knew separately that male mice are more prone to getting obesity and insulin resistance from a high fructose diet, but female mice are for some reason
Yes.
resistant to that?
Exactly. Yeah, that's
Really?
exactly right. Yeah.
Well, that's a subject for a different day, perhaps. But
Yeah,
with that in
exactly. Yeah.
mind, where do we go next in this study?
Yeah. So yeah, then the question is how does this happen? What is it that's what's actually driving the growth of the tumors? And we started to develop this idea that since when you sequence all the different tissues in the animal, and it's conserved in humans, you could do the same thing in humans, and you look at where the fructose is metabolized, you can see, as I mentioned earlier, and there have been precedents. I mean, there's other literature, some beautiful work out of Princeton, for example, that's shown the small intestine and the liver consume a lot of this stuff. But that's where all the gene expression is with respect to these enzymes that are required. So it becomes sort of obvious that somehow the utilization of fructose over here is driving tumor growth over there. And how does that happen? And we alluded to this earlier, that what can happen is that the healthy cells can output a nutrient, and that nutrient can feed cancer cells in a way that drives their proliferation. And so the model that we started to develop was that although fructose can't be utilized by some tumors directly, I do want to put a little asterisk on that, because if your tumor happens to be in the GI tract,
Then it can.
that's a caveat, and we can go back to that later. The tumors we're studying are like in the cervix and in the skin cancer and that sort of thing, breast tumors. So not in the places, the tissues of, and this is actually the question you asked earlier, tissues that originally have the capacity to metabolize fructose.
Yeah.
These are tissues that normally don't metabolize fructose,
Yeah.
where the tumors are. So the idea is that they're recruiting some nutrient that's derived from fructose, and that's actually driving the accelerated tumor
And
growth.
so this would explain the mystery that you mentioned earlier of the cells in the dish can't seem to use the fructose, but somehow the tumor cells in an animal are benefiting, being able to grow faster from fructose being present. So the idea here is certain cells in the body are metabolizing the fructose, not the tumor cells. That's producing something that then the tumor cell is somehow getting a hold of.
Exactly. And if we take, we guess the liver, because that's a big site of
Natural choice, yeah.
Yeah, fructose. And so what we did is we took liver cells, hepatocytes, and we cultured them in a dish, and we gave them fructose, and then we let them soak in the fructose for a couple of days, and then we take the media so they're taking up the fructose, using it and spitting stuff out, and then we took that media and we gave it to cancer cells, and as it turns out, cancer cells will grow on that. So that supports the notion that you just said, that the cells that have the capacity to utilize fructose take it up, transform it into X, and then X is given to the tumors, and X is a very good. Fructose is not a good nutrient for the tumors, but X, whatever that X might be, is.
Wow.
And that was really the hunt, is how do we find X? And that's, the other thing I'd like to just point out, because we were talking about this earlier, and this might be an appropriate time to insert it. You mentioned the word microenvironment, and I think that was a really nice summary that you gave, that the tumors are not just cancer cells, they're a whole aggregation of a bunch of different cell types. And over the last couple decades now, really, largely in part advances in spatial biology, people have gotten really interested in what's happening in the tumor microenvironment. That's really essentially that immediate environment surrounding the tumor.
Yeah.
And as we discussed, cancer cells manipulate other cells that are part of that. So even though they're, by all practical means, definitions are healthy,
Yeah.
we don't call them
Yeah.
fibroblasts. They're actually called cancer-associated fibroblasts,
Yeah.
CAFs.
The cells that are being extorted by the cancer
exactly.
cells.
And that's happening in that microenvironment. And so as we talk about this study now, we go back to the study that we just talked about, and we're talking about the liver, let's say, metabolizing fructose and maybe feeding a tumor in the breast. That is not the microenvironment. Those are now cells that are removed. They're on opposite sides of the animal. So we've been referring to that, as others has, as the macro environment. So it's not only, I think what we've moved, if we kind of look at the history of cancer metabolism over the last 100 years, we started off by just thinking of the cancer cell and the oncogene and what happens in the cancer cell. Then we sort of came to the appreciation, well, it's also these cells that are immediately surrounding in the microenvironment. And now we're saying, well, it's actually not just that, it's the whole system, the whole animal.
Yeah.
And just one last point that I want to make, and I think, in my opinion, this is one of the most fascinating observations that we've seen. If you take an animal, a healthy animal, and you transplant a tumor in it, and you compare that to a healthy animal with no tumor, and you compare the tissue, let's say let's add skin cancer. Let's give him a melanoma. We know that the melanoma, we talked earlier, that the melanoma lights up because it takes up a lot of glucose, and that's how we diagnose cancer in the clinic and stage it with PET. That definitely is the case. And people have really focused on the weird, funky metabolism of the tumor and the adjacent cells. But what we find is that almost every cell that we can find in the animal with the tumor is screwed up. It's not just, we tend to think of
the tumor is the epicenter of the cancer, but there's
the epicenter. But it stretches it spans the entire organism.
It's like an atom bomb goes off, but the radiation effects, can extend for miles and miles.
It's exactly the right analogy. So it's a systemic disease.
Yeah. Yeah. There's a focal point, but it is a whole body issue. It's
a whole body disease. And when you look at, I think, some of the things that when I look at where the field is going, I think you can see an increase in appreciation for this. And I think as our discussion matures here in the last half of our what we're discussing, I think that'll be interesting to come back to what implications that has, because I think that's going to have big implications of how we think about diet.
Yeah. Okay, so anyways, fructose can't be used directly by these tumor cells, but other cells are metabolizing it. It's turning into this mystery thing, X, and somehow that's assisting tumor growth. So fructose is indirectly facilitating tumor growth. Did you figure out what that mystery intermediate was?
Yes, we did. It's at least one of them. There's probably multiple mystery nutrients, but at least a major player is a nutrient called lysophosphatidylcholines. That's I know a big long word that probably a lot of people haven't thought about before. But that's a type of lipid, and it's unique. It's actually the what's happening here is incredibly interesting in the sense that it doesn't, at least for us, it was surprising when we think about what happens with fructose. You mentioned something earlier, de novo lipogenesis. So what happens often when you consume excess amounts of fructose is that your body, specifically your liver, converts that into lipids, and then those lipids get packaged together in a type of fat called tags, and they get excreted from the liver as a lipoprotein particle that floats around in the blood. It's like VLDL. You go to
all the stuff that we think about when we talk about blood cholesterol and LDLs and stuff.
Exactly. So most people are familiar with that because you get it measured, and there's good and bad and all that kind of stuff. So a lot of the lipid goes that's the destination. But what we found, irrespective of if they have cancer or not, but what we found is that in addition to making those kinds of lipids, fructose causes the liver to excrete this other kind of lipid called these lysophosphatidylcholines. And lysophosphatidylcholines are different than the other lipids that are packaged in the LDL, VLDLs, in the sense that they're a little more water soluble.
Okay.
So when you think about the lipoprotein particles, it's a very complex biochemical mechanism because you put all these
Mhm.
because the thing that makes
It's
lipids
a
is
way
they're
for the body to
insoluble.
move non-water soluble fatty stuff around in the watery environment of our blood.
Exactly.
Yeah.
So you build this huge particle and you
Yeah.
stick everything inside
Yeah.
of it,
But
and
these lipids are more water soluble. They can just kind of go in the blood or whatever.
exactly.
Yeah.
Yeah. So they get excreted, and they can circulate without the complexities of the
Hmm.
lipoprotein particles. And
Hmm.
that's important because if you think, well, how does the cell use the lipids inside of those lipoprotein particles? It's very complex. You have to figure out either how to take the whole particle in.
Yep.
There's kind of, macropinocytosis type process into a
Yep.
cytosis process, or you have to chip away at them, open
Wow.
it up, open up the vault,
Okay.
away at
So
it.
I think I see where we're going here.
Yeah.
These other cells that are not tumor cells, they're metabolizing the fructose. They're creating these LPCs, these fairly water soluble lipids. Those can now just go through the blood and the tumor cells will suck them up like they would any other energy source.
Yeah, I mean, you need a transporter,
Okay.
and we're trying to characterize, figure out exactly how that happens. There's still a lot of work here to do, by, I think we've uncovered some really
Hmm. But
good
they've, whatever it is, they've
ideas.
got it.
Yeah, yeah, and so they're able to utilize these much more directly, than you could with the more complex machinery that's involved in the lipoprotein particles.
Hmm.
Which is amazing.
Yeah.
It's kind of a magnificent way for a cancer cell to get access to lipids faster, more efficiently, potentially.
Yeah.
Yeah.
Yeah. And what do you know yet what the tumor cells are using these lipids for that supports growth? Is it for energy or is it
Yeah.
for structural lipid stuff?
Yeah. Yeah. That's a great question. I think those are the two possibilities. Exactly. They can use the lipids as a source of energy, or they can use them to build other complex molecules. And what we find is that, at least in the scenarios that we've studied so far, that it's the latter, that they're using them to build other molecules. So if you go back to one of the original statements we made, that when a cancer cell is defined by an unrestricted capacity for growth, and every time it proliferates or one cell turns into two, it has to remake all of its membranes. That requires it takes a lot of energy for a cell to make palmitate, a lipid.
Yeah.
And if it can take up palmitate directly,
That's energy efficient.
that's much more energetically efficient because it doesn't have to build it.
Yes.
So that is primarily what we're seeing. Something like 80% of these go that route,
Yeah.
that are used for structural membrane
Mhm.
lipids.
So these lysophosphatidyl these LPCs that were, this was the mystery thing, the mystery X we were talking about.
Yeah.
Fructose can turn into that in some cells, and then the tumor cells end up using that after they obtain this from circulation. Do these LPCs have any relationship to dietary fatty acids? Are they made from a certain type of fatty acid like that?
That's a great question. And I think what I'm really excited about now is trying to understand when LPCs go up. So we've learned that if you consume high fructose diets, that
Yep.
your LPCs go up. And we can make that same observation in people. So you asked earlier, is this some artifact of giving the mice the weird, diets? We see the same thing in people.
Yep.
So if you were to start eating high fructose diets, and we measured your blood today, and then in 10 days or a couple of weeks after you did it, we'd see increased
Mhm.
LPCs in your
Okay.
body as
So
well.
we know that the LPCs go up in humans in response to fructose. So unless the cancer biology is completely different from the mice and the humans here, this implies the implications are pretty obvious.
Yeah, yeah, I think so. But I think where I was going with that is, I think another really interesting question, and this gets to your point. What other instances do LPCs go up?
Right, right.
Because it's not something that at least we've thought about. Like,
Yeah,
I don't know that so it is true, if you do high fat diets, you might predict that LPCs go up, but
yeah.
but
But what
it's
is high
actually not
fat?
the case.
One of the most annoying things in the literature to me on the food stuff is, high fat diet is just too vague a term. I guess where
Yeah,
my mind was going with this is, if fructose goes to LPC, goes to faster tumor growth, the pool of potential lipids that can be transformed into LPCs might depend, say, on PUFAs versus, one type of dietary fat. And so sort of like the potential energy of what the fructose can be turned into to support tumor growth might be limited by something like the dietary fat that comes into play here.
yeah. No, I think that's a really great question. A lot of the lipid biology for cancer is still pretty nascent, I
Yep.
think.
Yep.
It's we've classified lipids together as a whole. As I totally agree with you. What you just said resonates with me strongly. I wish you would review our grants and papers with that perspective.
Yeah.
Because I think lipids as a whole have been sort of broadly classified. Oh, lipids all do the same thing. But
Mhm.
when you screen, certain LPCs like 18-1 appears to be an LPC that's particularly important for these cancer cells. They seem to like they do better with 18-1 than they do some of the other LPCs. Now, exactly why that is, we're not exactly sure. I don't know. I think there's a lot of interesting biology associated with specific classes of lipids and decorations and rearrangements.
Yeah.
And I think there's a ton to be learned there. But your point is absolutely well taken. What does this imply for what lipids we should be supplementing our food with? Certainly, someone asked me once, what happens if you just consume LPCs? What impact
Mm.
does that have? Does that change things? I mean, we didn't do that experiment. We infused LPCs into the intravenously, and that impact. If you just intravenously, if you infuse LPCs in mice, the tumors will grow faster.
Yeah. Makes sense.
Yeah. But remember that when you put something in your body, we've learned from fructose, it doesn't necessarily mean that what you put in your body is what the tumor is going to see. I think
Right.
if I had to say one really big take-home message, it seems so obvious, but I think one really important thing is that when we tend to think about, at least kind of canonically, when you think about cancer, you say, okay, I'm going to put X in my body, whether it's glucose or fructose or whatever, because I'm eating a lot of it. And that means that the tumor is going to get that. So
Yep.
is it bad for the tumor to have glucose? Yes, I'm not going to eat glucose. And then the sort of this like simplistic reductionism that if I don't eat glucose, the tumor won't get glucose. And if I eat a lot of fructose, the tumor is going to see a lot of fructose. But it's much more complicated than
Yeah.
that.
Yeah. What the tumor sees is not necessarily what you see because there's all these sort of filtering mechanisms at different levels of regulation.
Everything gets transformed by a million different cell types. And so we have to think about, and I think that's what we think about the interface of diet and cancer. That's the next frontier, is trying to figure out not necessarily what the nutrients do to the cancer cells themselves. I mean, that obviously is important. If we don't know that LPCs drive growth, then we don't know to ask what diets increase LPCs. But I think what we need to ask is, how do diets influence nutrient availability?
Right.
And then does that change in nutrient availability affect the rate at which tumors can grow?
Yeah. Yeah. So if you had to sort of compress the takeaways from this study, so we went through lots of detail here, but sort of the overarching questions that an ordinary non-biologist might want answers to here, or want to know what the relevance is what do exogenous sugars, things like fructose, things like high fructose corn syrup, is this basically stating that if you're eating too much high fructose corn syrup, whatever exactly that means, that it has the potential to facilitate tumor growth?
Absolutely. I think that is a take-home message. Obviously, I can't tell you that we validated this and have tested it a million ways in humans. That's all the next step. We got to take it further. Everything that's in the paper you've mentioned is all in animal models. So, but I think, as you say, there's compelling reason to believe that this will translate into humans.
Mm-hmm.
And I can say that I think it's not going to hurt you if you consume less fructose. Let's say it that way.
Mm-hmm.
And there is a big possibility that it will benefit you if you have cancer and you consume fructose. There's a possibility it could hurt you, but I don't see any possibility that it would be bad not to consume fructose.
That's actually a good way of framing it, because people in the diet nutrition, it's so fascinating, but people get so factional and tribal and almost religious with it. I want to repeat what you just said. So there's certainly a lot of downside potential to eating too much fructose, whatever exactly too much means. There's probably no downside risk to taking it down and eating less fructose.
Exactly.
Okay.
Absolutely correct. And again, I do want to put the caveat that what we're talking about here is not, avoiding fruit.
Right.
That's not what we mean.
And is it fair to say that like when you think about it, and we're not saying avoid all fructose or avoid fruit. Part of what that means, to me at least, is whole foods are naturally limited in certain ways, right? They have a certain amount of fructose inside of them. They contain fiber and other things that will limit the rate of absorption. And our bodies, obviously by definition, animal bodies have evolved to extract nutrition from whole foods that are part of the natural environment and have been for thousands and millions of years. When we
Yes.
start adding things on top of that is where we can start to get into trouble.
Yeah, absolutely. I think that's perfectly well said. And so you don't, the other thing that we didn't mention is that fructose can change the way that other nutrients are metabolized.
Hmm.
So it's not just fructose that gets metabolized. Fructose gets metabolized into, we said LPCs. We know that for a fact. But the way the presence of fructose can actually change the way in which glucose is metabolized. So
Also,
so for example, more glucose will make lipids in the presence of fructose than if you're not consuming fructose. So you activate the de novo lipogenesis, and that routes more glucose to lipids than you would if you didn't have fructose present. So if you do
yeah.
everything is exactly equal amount of sugar, but in one case you have fructose, in one case you don't. So
Mhm.
maybe you have fructose and glucose and just glucose, but everything's equal. You will make more lipid. Glucose will make more lipids when fructose is present than it does when it's just glucose alone.
Yeah.
So it raises yet another complication as we think about diet. It's not just what does this nutrient get transformed into and how could that feed cancer, but how does the presence of nutrient A affect the transformation of nutrient B? Because it could be that the presence of nutrient A makes nutrient B get transformed into something else, and that's what's feeding the. So it's
Yeah.
an extraordinarily complex situation, and I think that's why we just need more research in this space.
Yeah.
I think it's one of the more important areas, and maybe who knows, maybe with the current interest in the administration, maybe this is an area we'll see more resources invested.
Mhm, mhm. I mean, just to sort of reiterate or riff on something you said a moment ago, um, let me ask this question. What would happen if someone was consuming zero fructose? Would anything break? Would anything go
No,
wrong that we know about?
no. In fact, you can make fructose endogenously.
Yeah.
So if you here this is a little bit in the weeds, but it might help think about your question. So we developed as part of this study, we developed a drug that blocks the metabolism of fructose in the liver. So and it actually already exists. The drug we didn't invent it, we didn't design it or synthesize it. It was developed because it was developed in the context of fatty liver or steatosis, fatty liver disease. And basically the idea is, if we block the utilization of fructose, might that minimize fatty liver disease? Now, that went through clinical trials and it's not currently moving forward, but the drug was out there that exists that prevents the metabolism of fructose in the liver. And so we wanted to actually give that drug to animals and put them on high fructose diets and ask if we gave that drug, in theory, that should prevent the liver from making these LPC molecules.
Yeah.
And if they don't convert fructose into LPCs, then the tumors shouldn't grow faster. And that's actually what happened.
Hmm.
The tumors didn't grow faster. But kind of an unexpected, surprising result that we found is that when we administer the drug, in some scenarios, the drug actually reduces tumor growth more than if the animals weren't eating fructose at all. In other words, imagine that you get a certain tumor growth without fructose.
Yep.
And then you give this drug, which blocks fructose metabolism, but you don't give the animals fructose. So you expect to
So
get the
all of the
exact same.
fructose is endogenously produced.
Yes. So
Wow.
so you kind of stole the punchline, but
Sorry.
but that's okay. But why is it that the drug has an impact even when there's no fructose present? And we think that at least part of that reason is that the fructose is being produced endogenously
Yeah.
through a pathway called the polyol pathway that's present in cells. And
Wow.
but the reason I go through that big, it was a big long tangent, but the reason I mention that is because you ask how important is fructose? And if you don't consume it and for whatever reason your body needs it as a
Yeah.
glycolytic intermediate
You
or
can
whatever
make,
capacity,
yeah.
you can make it.
Yeah.
So
And not
you
only,
don't have.
yeah, not only that, but when I think about endogenous synthesis of things, Right. This is not, I don't want to make a blanket statement, but when your body has the ability to synthesize something from scratch, it generally, as long as you're otherwise healthy, will make just as much as it needs. So, for example, most animals, not humans, but many animals have endogenous vitamin C synthesis capabilities. So they don't need to worry about how much vitamin C they consume. Their body will make exactly what they need as long as they're getting all of the nourishment, other nourishment they do need. I would naturally think it's probably similar with a human being. If we can produce fructose endogenously, then we probably don't need to think too much about trying to get some.
Yeah. No, I think that's a good rationalization. And certainly with fructose, particularly because when you produce it endogenously, it's regulated.
Yeah.
As what you're basically saying is that
Yeah.
your body regulates it. So you tend to not have to worry about excessive production because your evolution tends to allow us to produce what we need. When you're eating, entire chocolate cakes, that's a situation where you lose the regulation component.
Yeah, yeah. And this is beyond the scope of our discussion, but obviously the tie-in with cancer stuff is what we just learned from you. But excess fructose consumption also, as we touched on, it can promote fatty liver disease, insulin resistance, potentially in a sex-dependent manner, and
Yeah.
lots of other stuff besides cancer.
Yeah, exactly. Yeah, and I think we're just scratching the surface for we talked about sugars today, but there's a lot of other sugars. We're interested in some other artificial sweeteners.
Yeah.
We're interested in it's not just fructose and glucose. There's if you go look at your favorite foods, that they're going to have other sugars in there as well. And so this question is interesting. The discussion we had today is quite interesting for fructose. Certainly, that's the most important. Just if you look at consumption rates, but there are other sugars that I think that we're learning are going to play an important role in the same context as well.
Mm-hmm.
Some other sweeteners that I think are going to be equally important and have similar types of effects. So, and some of these, I think, the scary thing with some of the other sugars that we're looking at is that some of these things are considered, you can imagine if you're on an Atkins diet or a non-ketogenic diet where you're trying to avoid sugars and use artificial sweeteners. But if the cancer cells can use those sweeteners or they can make these LPCs, that's not necessarily the way to go.
Mm-hmm. Mm-hmm. And so what would be so given this study and everything about fructose, cancer, all this stuff, what would be your take on questions that people are asking in society around high fructose corn syrup versus cane sugar, how much is too much and all that stuff? To sort of preempt you there, my sort of general take on all those things is, okay, high fructose corn syrups is 55% fructose, 45% glucose, cane sugars 50-50. I would say a 5% difference is a 5% difference. If it's in the context of eating 200 grams of added sugar a day, that's probably not meaningful, but a 5% difference is a 5% difference. It's going to nudge things a little bit one way.
Yeah. Yeah. I mean, I guess my take is, quantitatively, I believe what you're saying is that makes sense to me. I think the idea that if we get rid of high fructose corn syrup and move to cane sugar, that everything goes away, that all the issues that we've associated with fructose are going to be obviated. I think that is nonsensical.
Yeah.
But you're right. It's not exactly the same. So you might see some biochemical differences because quantitatively there's subtle differences in the fractions. But I think the idea that just moving away from high fructose corn syrup and going to cane sugar is going to eliminate the phenomena like what we saw in this paper and associate with liver disease and obesity. I think that's very unlikely.
Mm-hmm. So maybe another way of saying that would be, if you're drinking a liter of American Coke a day and you switch to Mexican Coke, probably not going to move the needle, especially if nothing else in your life is changing. But if you were to switch to Mexican Coke and cut down the volume you're consuming, that could make a difference.
Absolutely. And I would say cutting down the volume is much more important than the cane sugar or fructose.
Mm-hmm. One of the difficulties preparing for this was you've done so much that it was difficult to pick exactly what to talk about. But I definitely wanted to talk about this fructose paper. What else, just in the realm of cancer biology in general, can you tell us about, say, dietary sugar versus dietary fat? And I want to keep it, we've got just a few minutes left. I want to keep it pretty broad. Earlier, you mentioned, for example, this notion of Okay, well, cancer cells are growing it's unchecked growth, so they're very metabolically expensive. They have to suck up and use a lot of energy. If they like to use things like glucose for energy and they suck up a lot of glucose, it makes sense to deprive them of glucose intuitively as a way to try and treat or slow down the tumor. That would naturally bring us to questions of like, okay, if someone has a tumor and we put them on, say, a ketogenic diet, is that known to have any kind of effect on tumor growth?
Yes, it's a great question. And I think very empowering to people who have cancer, because it's something you can control. The idea that you could change your diet and that might have an impact on the course of your disease, I think is a very enticing idea. And I'm very optimistic that that is a possibility. Certainly the example that we outlined with fructose is maybe a good starting point, right? If you consume more or less fructose, it's likely to impact the course of your disease. Again, with the caveat that we need to do more studies in humans to say that definitively. But the idea that you could do a ketogenic diet and quote unquote starve the cells of glucose has not worked. And that's something that people have thought about for a long time. But you can imagine decades ago, it's not such a big leap conceptually to say, if we know cancer cells are addicted to glucose, what happens if I stop eating glucose? And it really does get at this idea that we chatted about a couple minutes ago, that you have to think about this in the physiological context. If we had cells in a dish and we had media and we just put the cells and we said we're going to take glucose out of the media, yeah, that might work. But we mentioned earlier that glucose is regulated in our blood with exquisite regulation. So, if you don't eat glucose, your blood glucose doesn't drop. I mean, it drops a little, but it has to be maintained in a certain range because your brain needs glucose. And so you can't, just because you stop eating glucose doesn't mean that glucose disappears from your blood and that the tumor doesn't have access to it. And what's really interesting, so maybe I could just mention briefly another study that we did that's highly related to this. So what does happen if you stop eating glucose? Like, let's say, because it's a really interesting question to think about, right? If you're not a metabolism person, like let's say you don't eat for 2 days, and I just said, but your glucose levels are maintained. Like, how does that actually happen? And the way it actually happens, it goes back to the liver, which in many ways is the kind of control center of metabolism in the body. Your liver makes glucose. And kind of what you were saying earlier, like if you need something, you make it.
Yeah.
And so your liver will take other things in your body and make glucose out of them. And so that's how you maintain your levels of glucose in your blood.
This is the process of gluconeogenesis.
Gluconeogenesis.
So you can take a chunk of muscle and turn the protein into sugar if you need to.
Exactly. So anything that's glucogenic, you can turn back into glucose. You can't turn everything into glucose, but a lot of stuff you can. And so what ends up happening is that when you have tumors, what we found is that when you have a tumor, tumors take up tons of glucose. So normally in your body, and hopefully all of our listeners' body, that gluconeogenesis only happens when you're deprived of you haven't consumed glucose.
Yep.
But what happens when you have a tumor is that gluconeogenesis is happening constitutively. It's always happening, even when you eat, because the tumor is consuming so much glucose.
Yeah.
That it starts to lower the level of glucose in the blood.
So it's your body's, in a sense, it's like your body's starving, even though it's not starving.
Yeah. Exactly. Yeah. Yeah. So the glucose starts to drop, because the tumor is consuming the glucose, and your body says, well, I got to keep the glucose levels high, within this range. Yeah. And so it starts making it in the liver. So that kind of illustrates again, and that's not a tumor in the liver per se. That's a tumor anywhere in the body.
Yeah.
But it's again this physiological response that's happening. So I think that's really pretty fascinating. And maybe just one other thing I'll comment on in connection to that. When we think about the whole body, I've been really emphasizing that cancer is a whole body disease. And from a metabolic perspective, there's another thing that happens when patients have cancer that represents that probably most emblematically. And that is a phenomenon called cachexia, cancer-associated cachexia. And it reminded me because you said you can take a piece of muscle and turn it into glucose. And what happens in cachexia is that patients will start to lose body weight. So they'll start to lose fat, and they'll start to lose muscle mass. And in many cases, the reason that a patient dies from cancer is associated with cancer cachexia, not because of the tumor per se, but often
Hmm.
it's because of the cachexic response.
They just
And that's
waste away,
you
basically.
waste away. Yeah. And that's basically what's happening. So it doesn't matter where your tumor is. I mean, you could have a pancreatic tumor, but
Yeah.
your muscle and your arms and your legs and your fat tissue around your body gets dissipated.
Hmm.
So that's, when you said that, I think metaphorically, kind of whimsically is maybe a joke, but that actually is not so far away from what actually
Right.
can happen.
Yeah. Yeah. Wow. Yeah. Your body, so the tumor is just using so much sugar that your body thinks it's sugar deprived, even though you're maybe eating an otherwise normal diet. And you will quite literally start breaking down your muscle and turning it into sugar
Yeah, exactly.
to feed the tumor.
Yeah, yeah, 100%.
Wow. Um, is there any connection between um, constant nutrient and caloric surplus and the probability of getting cancer? So another way of saying this, if cancer is unchecked cell growth and cancer cells are always popping into and out of existence, but we normally detect them and then get rid of them, but sometimes they can take a hold and the tumor will grow and all that
Yeah.
stuff will happen. Is there any reason to think that are cancers more likely to stick and turn into full blown tumors in a state where you always have calories around, you always have surplus nutrition, you're never fasting, you're never cycling in between, say, ketosis and non-ketosis? Is that a risk factor itself, just always having surplus energy?
Yes, absolutely. I believe so. I think the evidence is out there to extrapolate from. And there's a couple reasons it's a little bit of a convoluted question, but I will tell you personally that I do fast. And for 7 years, I fasted where I only ate 2 hours a day.
So
For
you're a
this,
daily intermittent faster.
yes. Um, but the to get to your question perhaps more precisely, that one of the reasons it's complicated to answer your question is if you're constantly in a state of excess calories, you become obese. And we know for a fact that obesity is associated with increased risk of certain cancers.
Mm-hmm.
Now, exactly why that is a complicated question, and something that we're trying to sort out. As I mentioned earlier, we do know that cancer cells prefer to localize next to in pockets of fat. Exactly why that is it because they're getting extra nutrients? The fat can, there's signaling going on. You mentioned earlier about different kinds of signaling lipids that may be a role there. So we don't know exactly why that happens, but it's absolutely the case that that obesity does increase your incidence for contracting certain kinds of cancers.
Hmm. Interesting. Yeah, and I mean, yeah, it makes sense. I mean, it just makes perfect sense. So you do daily intermittent fasting.
Yes.
Interesting. And so is there any reason to, I mean, this might be too much detail, but is there any reason to think about, do you think daily intermittent fasting has benefits above and beyond intermittent fasting? Like if you're just fasting, every couple of months or something like that for a day or two, does the pattern and tempo of the fasting make a difference? Is there any literature on that with respect to cancer?
Yes. I don't. One of the challenges that we have, we've done quite a bit of experiments in this space. One of the challenges you have is that animals, we use a lot of mice and we use a lot of zebrafish as model systems. And one of the challenges that you have is, let's say that you fast, a fish for 10 days. What does that mean? Like, how does that compare to how we fast? So I could tell you exactly the fasting intervals and how they impact the fish, but
Mm-hmm.
how to translate that into humans, it's much harder to do that. And I think that's why, my clinical colleagues that are moving more and more towards doing clinical trials with nutrition is going to be so key.
Mm-hmm.
And hopefully the animal work that we've
Mm-hmm.
done, we and many, many others can provide hopefully some ideas
Mm-hmm.
that hopefully we'll be able to test in the clinic. But the answer is yes, you
Yeah.
can change the intervals of these fasting periods, and they do have an impact. And what you'd like to do is to try to make it the most palatable as possible, right? So
Yeah.
what's
Yeah.
the minimum amount of what's the least amount of times that you can fast for the least amount of hours? And that's what everybody will do, right? If you only have to fast once every 6 months, that's what we should do. But or at least maybe that's what one would choose to do. But we don't have that worked out yet in humans. Like I said, maybe we have some data that would be supportive in animals, but trying to correlate that is challenging.
Yeah. But I guess the punchline here is it sounds like what you're saying is that in animal models, in some animal models, fasting does affect, doing some amount of fasting will decrease the odds of cancer, and the exact way that you do the fasting does have some effect.
Yeah. And I also want to be clear here that what we're talking about here is not if you have cancer. So if you have cancer and you try to fast, that is different. You got to this point earlier, than actually initiating cancer. So there's a difference there. So and fasting is not equal across both of those paradigms. So if you have already a tumor that's been clinically detected, fasting there, the evidence is relatively weak that it has much of an impact. Now, it might have an impact when combined with certain therapies. I think that, but there's so many combinations, it's hard to, that there's a lot of possibilities to test there. Or perhaps fasting, there's some really nice work out of Duke, for example, that shows that maybe you don't have to fast everything. The beauty of the ketogenic diet is you can eat as much as you want, you just can't eat glucose. And it might be, well, you can eat as much as you want, you just can't eat this essential amino acid. And there's some work coming out like on methionine and things like that. But there's a difference when you already have the tumor. And I think the dietary strategies that will emerge if you already are unfortunate and you have cancer, this is what you should do, versus you don't yet have cancer. What dietary practices should you use to limit your chances of getting cancer? And kind of circling back to contextualizing this with all fructose. For example, our study showed, and this is something that people often ask that I think is not our intent. And if we communicated it that way, it was a mishap on our part. But what our study shows is that if you already have a tumor and you eat excessive fructose, it will make your tumor grow faster. It does not say that if you do not have cancer and you eat fructose, that you're more likely to get cancer.
Yes.
Those are very separate things.
Yep.
Because as you pointed out earlier on, and it was a perfect way to start our conversation, the process of initiation is different biology than the process of the tumor development and progression. Yep. And so those are different phenomena. So what we're talking about here in the fasting is the former, that initiation process, versus the progression and development.
Yeah. Yeah. I would actually, my naive prediction would be that if you already have a tumor and you start fasting, well, because the tumor is good at sucking away energy that your body would otherwise need, you might just promote wasting of your healthy tissue.
Exactly. And cancer-associated cachexia, as I mentioned, is a big part of the demise.
Yeah.
And one of the things that people have worked on is how to limit that. And one way to potential strategy to limit that is to infuse nutrients, to put people on excess nutrients, to prevent that from happening. So I think you're right. That is certainly, there's some complexities there. But I think that maybe the take-home message is that cancer biology is complex. There's different phases of it. And depending on which phase you're in, you really have to contextualize the therapy to that particular phase. So that's the key point, I think.
Yeah. Well, I think that's a great place to end it, Gary. Thank you very much for your time. This was fascinating. This is one of the best ones I think I've done in a while. And yeah, I would recommend everyone who's interested in metabolism and cancer generally check out Gary's work, because you've got a voluminous set of things that you've investigated here, way more than we can get into. But the new paper on dietary fructose I thought was particularly interesting and very well done. So thank you for your time. And do you have any final thoughts you want to leave people with?
Yeah, no, I really appreciate it. It was a lot of fun. And I just shout out to the first author of the person who did the study, Ronnie Fowler-Greider, who led all the work and deserves all the credit for everything that we discussed today.
All right. Gary, Patti, thank you for your time.
Thank you.
Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. Metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.